Enzymes are omnipresent in our everyday lives, such as in laundry detergent for removing stains, and are important for brewing beer. But their most important functions are in human cells. Enzymes catalyze almost all chemical reactions in a cell's metabolism, causing the reactions to proceed much more efficiently than they would otherwise. Phosphorylation is an important chemical reaction in our cells, affecting our proteins. In phosphorylation, a phosphate group, such as this one, is transferred to an amino acid in the protein. The cell has a large reservoir of phosphate in the form of ATP that it can use for phosphorylation, but transferring a phosphate group from ATP to the amino acid is a slow process. So enzymes come to the rescue. A kinase is an enzyme that can make phosphorylation proceed much more rapidly. Enzymes work by having a three-dimensional shape that exactly matches the shape of the molecules they need to recognize. In enzyme language, these are called substrates. The three-dimensional shape keeps the substrates in place relative to each other, such that the reaction does not require as much energy and proceeds faster. The meeting between enzymes and their substrates has traditionally been considered similar to a lock with a key. The substrates, the key, exactly match the shape of the enzyme, the lock. And just as there is only one way to put a key into a lock, through the keyhole, the substrates only fit in an enzyme in one way, the active site of the enzyme. But in recent years, research has shown that the meeting between enzyme and substrate is not quite as simple as just a lock and a key. New research in Denmark has investigated this by examining the mechanism that enables kinases to recognize their substrates. Kinases need to distinguish between similar substrates. They therefore use docking interactions, such as two alpha helixes, that are not located in the active site. If the substrate is the key that fits a lock, then the docking interaction directs the key to the right lock by grabbing the door handle. These docking interactions often comprise unstructured proteins with a long attachment called linkers, and they are not rigid structures like the enzyme is. The linkers look more like a cooked dancing spaghetti. The strength of the docking interactions and the length of the linkers affect how efficiently an enzyme processes its substrate. The strength must be great enough for the enzyme and substrate to come into contact with each other, and the linkers must be able to lead the substrate into the active site of the enzyme. Only then can a reaction occur. But the interactions must also not be so strong that the substrates are not released again after they have reacted, because then they take up space and new substrates cannot be added. That is not good either. The research group in Denmark investigated the enzyme's efficiency by mutating the docking interactions and thus changing their strength, and by changing the length of the linkers.
We have developed equations that predict the optimal binding strength between an enzyme and a substrate. Our conclusion is that the interactions should be of intermediate strength, and for the first time we have actually managed to quantify what medium strength means.
The researchers' equations can thus be used to understand the effect of specific mutations in enzymes and their substrates. But this new knowledge can also prove extremely useful for designing new drugs.
There's a new and very promising class of drug molecules that work by targeting certain proteins for degradation by sending them to the garbage can of the cell. This could, for example, be a protein that's involved in the development of cancer. Our equations describe the theoretical foundation for optimizing such drugs, and we're very excited about the possibility of actually getting to test this in the lab.
You can read more about this research at sciencenews.dk.